I want to thank you very much for coming today, and I'm extremely honored to be here because you're the future of medicine, and I'm at the University of Washington and Seattle Children's Research Institute, and when Dr. Das asked me to give him a topic that is important for medicine, I felt like I want to give you the topic on personalized medicine. And often people call it not personalized medicine, but precision medicine. And I think all of this is kind of a synonym of where the future will be. And my own research and my institute is really dealing with neurological disorders. And it's a big problem. And just to give you an idea how big of a problem it is, in India, brain tumors, like you have 5 to 10 cases per 100,000. So 500 cases of brain tumors are diagnosed every year in India. You have 6 to 10 million people in India that suffer from seizures. And you have also a sleep apnea problem. So 12.7% in India suffer from sleep apnea. And in fact, this will rise very dramatically because it's related to obesity, and you have huge problems with diabetes here. So I predict that also sleep apnea will be a dramatic problem, and how can we deal with this? Then we have a lot of psychiatric disorders. In the US, we have 150 children suffering from autism, and it's going up, so there's a 470% cumulative growth rate that since '92, and we think that in India you have 12,000 per year that are diagnosed with autism. Then of course you have major depression, anxiety. We have a huge problems of addiction, and in the US, people think that 47% of the US population is afflicted with addiction, and we have a major problem with opiate diseases. So you can see there's a lot of neurological disorders that we're dealing with, and at the moment there's a big problem because we cannot cure neurological disorders. We can treat them, but there's no cure. And so right now we classify disorders very roughly. We say you have epilepsy, and then we try to give these epileptic patients an anti-epileptic drug, but really the population of epilepsy patients is very heterogeneous, and so many drugs that we give help initially, but after a while they don't help, and then we have a population of patients that have epilepsy but we cannot treat them, and we call this the intractable epilepsy patients. And in fact it's like 30% of the people with epilepsy are not treated and become intractable, and even if we control them initially with anti-epileptic drugs, it doesn't help. And this schematic here is a general schematic which not only applies to epilepsy but really also to autism. Right now we have a lot of autistic patients, and really it's like a trial and error how we treat them, because autism is many diseases, and every patient with a different autistic disorder should be treated differently. And for us, in the research arm, what we realize is we have to optimize, we have to better understand, what is in this bucket of a neurological disorder. And we have learned a lot from cancer research. And in cancer research, for many years, people said you have breast cancer, you have brain cancer, and you have ovarian cancer, and or you have a blood cancer. But a major breakthrough in cancer research came when we started to say you don't have any more breast cancer, but you have a breast cancer that is related to this and this receptor and through this and this mechanism. And then we targeted the mechanism, and as soon as we target the mechanism, we have success in treatment, So basically there's a huge effort in not saying you have epilepsy, but saying you have epilepsy because of this and this reason, and because you have this and this reason, we give you a personalized treatment in order to treat your epilepsy. Okay, so you see, it's a general concept that you as a clinician have to learn to think mechanistically, not only based on the symptom, which is in this case epilepsy, but it could be also autism, it could be etc. Now, one approach, of course, to get to personalized medicine is the genetic testing, but genetic testing is not so easy, and it's not also giving you the right answers often. And one example is, for example, ADHD, attention deficit disorder. And we know that there are many, many genes involved. You know, like you have some problems with the dopamine transporter, some genetic mutation with noradrenergic transporters, serotonin transporters. But there are many genes that can lead to ADHD, but each of these genes has a very small effect. And it really causes only a predisposition. So if you know that someone has an ADHD and you identify a mutation, it really doesn't tell you much because the mutation doesn't play a big role in the picture. And what we've learned that in many cases, it's actually the environmental condition together with the predisposition that lead to ADHD. And so we have to identify, you know, what are these underlying environmental factors. A lot has to do with maternal stress, psychosocial stress, and that basically together with the gene can lead to ADHD. So what does it help us now? If we know that there are genes there, we know that there's a stress that leads to ADHD, but how does it help us in our treatment? And it helps us because if we understand how this environment, how this gene affects brain development or brain activity, then we can start to develop ideas how we can target these mechanisms in order to treat ADHD. Right now, what we do is we just give like psychostimulants without really understanding the mechanism. Now, so what we need is not only to understand the genes, we need to understand all the mechanisms from the environment to the genes to the neural mechanisms in order to then treat a neurological disorder in a much more systematic way. And what I want to present you is some examples from our center. Because our center was built, we started it in 2009. And in fact, I started it, and the vision was that we bring people together, experts together, that are experts in human genetics, that are experts in molecular biology, that are experts in cell biology, that know neural networks, that know behavior, and that also understand environmental factors. And if we bring a team together like this, it's a lot of young people, as you can see, then you can actually go and understand the mechanism, you can develop therapies, you hopefully also can develop cures in order to treat these neurological disorders. So I will tell you a lot of stories, for example, from Reda Mirza. She's a human geneticist. Kathy Miller is a mouse geneticist. And Teresa is, for example, a person that can manipulate genes, so she can develop genetic therapies. And we have a team of molecular biologists, etc., that all work together in order to create new ideas. And these are the faculty. So we have approximately 20 faculty, but really what I show you here is only the tip of the iceberg, because we have, these are the scientists, the research scientists that sit in our building, but we have also another 40 people that sit in the hospitals. So we have basically these hospital scientists and the clinician scientists, and then these basic scientists that actually look at many different disorders. We have specialists in cardiology, autism, autonomic dysfunction, brain tumors, eating disorders, metabolic disorders, impulse disorders, and also epilepsy. And we have these researchers that use a lot of different technologies, and you can see that every researcher uses different types of technologies to get at the precision medicine, personalized medicine. And I decided today to give you some insights more on epilepsy, but I could do the same thing for mitochondrial diseases, for brain tumors, for autism, but I want to give you some on epilepsy. But before I go into epilepsy, I want to give you also some ideas how we tackle autism. So in autism, we have a huge problem that it's a spectrum. Okay, there's not one autistic disorder. It's many, many disorders. And there are many, many genes that lead to autism. And what I've heard is that if one psychiatrist sees thousands and thousands of autistic kids over their career, the likelihood that this doctor, this psychiatrist, saw one kid with the same mutation twice is very, very low. So basically, if you see 7,000 autistic kids throughout your career, the likelihood that 2 had exactly the same mutation is very low. So that is a big problem. So basically, how do we go about this? We have to go about this personalized. And we have several people that actually focus on this. One is Steve Smith, who is a young guy. So he's like 40, and he's a scientist. And what he does is he takes autistic kids and takes the hair of the autistic kids. And with this hair, he turns this hair into nerve cells. And then he turns these nerve cells from different autistic kids and asks, first of all, what kind of mutation do you have? And then he finds that many of these autistic kids have problems with the glutamatergic synapse. But knowing that it's a mutation with the glutamatergic synapse doesn't tell you much. So what he does is he studies how they affect the proteins of the glutamatergic synapse. So he's studying protein-protein interactions to see, whether this autistic child has certain protein-protein interaction problems that are similar to other children with this mutation or with different mutations. And then basically he can start getting ideas, what kind of autistic kid belongs together mechanistically with other families with autistic kids. Then we have another person, John Welch. He also looks at autistic kids, but he looks at autistic kids in a different way. He looks at the EEG. So he uses electroencephalographic recordings from autistic kids. And why does he do this? And the reason for this is that basically all our neurological disorders have to have problems with electrical activity. And what we see very often in autistic kids is that they have problems in processing time. And so what he developed is strategies to test the learning and EEG activity of these autistic kids. And what he finds is that there are different types of autistic kids. Some which process information faster, and some that process information slower. So if you process information faster, then you have a problem because it's very difficult to process for example, a face, or to process a human interaction. So for someone with autism that has problems with the processing, then looking at a face is very scary. That's why they want to look at an object, because an object doesn't move, it's very well defined. You look at a chair, no problem with a chair, but looking at a face, there are many, many signals and they cannot process this enough. So what he does is he devises learning devices to test, is this person processing too fast or is this person too slow? And then you can train these people, okay? You can train the people to start processing faster information or slower information. Also, depending on how fast they process auditory information versus visual information, tells you where the problem sits, okay? So he starts to say, this person has processing slow in, let's say, social interaction, this one in visual interaction, and then he can categorize these different patients based on different types of autistic spectrum disorders that are described by processes. in the brain. And he can train, and in fact, he started with rats. Because in rats, you can also make mutation that process time differently. And he can say, okay, you have probably a basal ganglia process problem, you have a cortical problem, you have a cerebellar problem. And then he can train these people, and the earlier you discover this, you better can train people to strengthen their connectivity, to strengthen this. So this is kind of an approach to look at autism with EEGs, etc. Now, I wanted to talk more now about epilepsy. And we always say a person has epilepsy because they have certain seizure problems. So they get synchronized activity. But really, there are many, many reasons why you can have seizure disorders. Very similar to autism. You can have many, many reasons why you have problems with social communication and social processing. And so what do we do about this? And there has been a lot of progress in the last few years in identifying the genetic basis of epilepsy. And this is Heather Mefford. She's a colleague and she's also part of our center. And she has been part of this human genetic revolution. And I call it a revolution because we have really have an explosive understanding of the genetic basis of epilepsy. So unfortunately, we don't see the year here, and I can't read it here, but let me see. So basically, on this x-axis is 1995 was the first channel that was identified as a genetic mutation that can lead to seizures. And this is 2005, so we had already several genetic mutations that were identified and linked to epilepsy. So one of them is SCN1A mutation. It's a sodium channel mutation that leads to something called Dravet syndrome. So these are children that have seizures very, very early on, and many of them actually die because they have something called sudden death of epilepsy. But you see there are also mutations in the GABA channels that are like these, GABA A1, etc. There are certain mutations in potassium channels. And this was all discovered in this area, in this time frame from 95 to 2005, which is like a 10-year space. Now, this channelopathy area was then replaced by the so-called next gene sequencing area, next generation sequencing area. Because nowadays, it costs only like $1,000 to do a whole genome of one patient. So now this allowed us now to test way more children and that identified many, many more genes that are associated with epilepsy. And this basically was done by new ways to do genetic testing. And one of the ways is the so-called trio mutation, trio analysis. So what you look is, you take a patient that has epilepsy, you do the sequencing of that patient, but you also look at the sequence of the mother and the father, and then you compare the sequences. And what it allows you is to identify spontaneous mutation, and in fact, most of the epilepsies are due to these de novo spontaneous mutations. So because you have the parent sequence, all you have to do is find now a mutation that is different from the parent. And that allowed us way faster to identify a mutation that is different, and many of the human mutations of genetics in epilepsy were identified by this trio approach. But then of course you have also the dominant heritage, so you have also families that inherit it, but as I said, this is not the biggest one. Now, once you know the gene mutation, this is only the beginning of a personalized medicine journey. And because you have to understand exactly where this genetic mutation sits on this gene in order to get functional insights. And I want to give you an example from our own lab, so my lab. And this is work done by Adhd in my lab. He studied all the time potassium channels throughout his life. He looked actually at worms and then in mice, etc. And he found that if you mutate a channel called casein Q5, it's a potassium channel, that we made these mice with this mutation, and these knockout mice had seizures, okay? So that told us that if you have a mutation casein Q channels, you should have seizures. But this was done in mice, and if I would have been at a university where there are not human geneticists, that would be the end of my research. I would have published that casein Q5 causes seizures in mice. But at Seattle Children's, because we have this research institute, we went next door where Bill Dobbins sits, who is a human geneticist, and said, hey, you know what? We have mice that have casein Q5 mutation. They cause seizures. We predict that there will be patients out there in the world that also have seizures because of this channel. So because he's a human geneticist, he can have databases where, this is, I don't know what I want to tell you. Basically, he immediately identified several patients throughout the world, in England and then in Holland and in America, and we identified 6 patients that had mutations in this casein Q channel. And this basically tells you exactly where this mutation was located. And then once you have these mutations identified in patients, what you can do is you can use a computer model. Okay, so there's a model that then Adhd can use, and that computer model tells you exactly where in this ion channel of the casein Q5 these mutations are sitting. So there are patients here, one patient had a mutation here, one patient has a mutation here, and now Adhd is an ion channel person, so he knows exactly where, what function each of these domains have. So he could tell, well, these mutations affect the pore, this mutation affects your voltage sensor, etc. And then you can have some functional ideas of what that mutation does to the channel of this potassium channel. But again, this is only the beginning, because now comes the question, what actually does this mutation? And so for this, you can use oocytes, so you can use HEC channels. So now you use the CRISPR approach and then insert for each of these patients this mutation into your channel and into a HEC cell, and then you can record, and what he finds is that these mutations here, they all cause a stronger expression of this channel. In fact, it was so-called a gain of function mutation, whereas 2 of these patients had a loss of function of the potassium channel. So what does it mean? That was a surprise to us that in fact most of the human mutation had a potassium channel that worked actually much stronger. So it was hyperpolarizing the cells at a much lower voltage than a normal channel. So they had actually more inhibition than normal, but these patients actually had a lot less inhibition. So what it tells you is that the same channel mutation can cause a gain of function and a loss of function, and both causes an epilepsy. So this has, of course, now clinical implication, because now both of these patients have a mutation in casein Q5. This has a loss of function, this has a gain of function mutation. So what it means is if we want to treat the epilepsy of this patient and this patient, we need actually the opposite kind of drug. Okay, so that is something for you clinicians very important. Okay, so because if you give a potassium channel agonist to this patient, you might treat the epilepsy. If you give the same drug to this patient, you will actually make the epilepsy worse. And whereas here, we think you need an antagonist to treat this patient, whereas here you would need an agonist. And again, the power of the personalized medicine will be now that there are casein Q agonists that we can test on these patients, because they could be used for blood pressure, for example, control. So what we now have is a personalized medicine approach where we can take a blood pressure drug to this patient in order to treat their epilepsy. So we would not use an anti-epileptic drug on this patient. So this tells you a little bit of how this personalized medicine can change medicine, because now you have a bigger repertoire of drugs that you can use that normally you would never use for this epileptic patient. But also it tells you why it is important, because this blood pressure medicine could actually kill this person, because it would have a different effect. Okay, so this is kind of the power of this. Now, for us, again, that's not where we stop. So the next step is we want to understand the mechanism. What kind of neurons are actually involved in this? And if we know that this potassium channel inhibits neurons, and this inhibition causes seizures, what we hypothesize is that likely this channel sits in inhibitory neurons in order to get epilepsy, because it means that if these inhibitory neurons get inhibited, you get excitation that then causes a seizure. Okay, so you have now a conceptual hypothesis based on this personalized therapy approach, and what we can now do is we can go into a database that is created by the Allen Institute. So the Allen Brain Institute has sequenced all the neurons and said, where, in which neurons are these KCQ5 channels located? And so this is then taken from the Allen Brain Institute, and so we know that this KCQ5 channel sits in these VIP inhibitory neurons. Okay? So this allows us now to make a very specific hypothesis that, in fact, these patients have an epileptic seizure because probably their VIP neurons are not working properly. So what we can do now is a mouse model where we can mutate exactly these VIP neurons, and our prediction is that they will have seizures, and that allows us now to have an animal model for that one patient that allows us now to treat this patient, find other drugs that actually target these VIP neurons in order to treat the epilepsy. So this is just one example, and of course the KCQ5 channel mutations are rare, but the Dravet syndrome is not very rare, and they are actually pretty frequent, these patients. And in this case, we have SCN1A that excites inhibitory neurons. And so again, now we know that you have basically here a mutation, it's a loss of function mutation in these neurons, these inhibitory neurons, and you can see that all the inhibitory neurons have the SCN1A mutation. And so what this faculty, Frank Kaluma, is now doing, he works at the Allen Institute to develop a therapy, a gene therapy, where he replaces the SCN1A mutation with another sodium channel to treat these patients. Okay, so basically, again, it's a personalized medicine. We understand the mutation. We know exactly in which neurons they're located, and that allows us now to do gene therapy on these patients in order to stop their seizures. So right now, I have to say that this personalized medicine approach is really at the research state. Okay, so I'm thinking that your future generation of medicine doctors, for you will be normal. Okay, for us it's not normal. I'm sure in India nobody will do this, and in fact in the US nobody is doing this. It's really just at the beginning of research. But when I think what we could do 5 years ago and what we can do now, it's an explosion. And so I predict that in 20 years, by the time you're at the peak of your career, this will be normal, okay? And nobody will give someone a drug without knowing why you give the drug. And the reason this is all developed is also a financial one. Like it costs $1 billion to develop, to sequence the first genome. Now it's $1,000. Now, if you have a patient with Dravet syndrome or with epilepsy, $1,000 is nothing to really find out exactly what is the cause of this patient. If you have an autistic kid, there is no reason anymore. For $1,000 you can know, does this have a mutation that then allows you to better predict what it is. Now, the problem is there's so many mutations. So these are all mutations that lead to epilepsy, and these are all mutations that actually alter GABAergic neurons to cause inhibition. So you can see, when we talk about epilepsy, without knowing this, we're really very brutally generalizing. And so the future will be to actually